Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Today we are going to have some discussion about prokaryotic and eukaryotic cells as well as the basic introduction of cell biology. You know, these are the contents of Animal Physiology, Zul 203, one of the chapter from cell biology. And the topic which we are going to cover from these contents includes cell biology introduction and major differences from prokaryotic and eukaryotic cells. Initial portion of cell biology, it has been taken from chapter number 2 of Miller and Harley zoology, and differences between prokaryotic and eukaryotic cells, they have been selected from various points, various sources. First of all, this lecture will cover introduction, ultrastructure of cell, as well as difference between prokaryotic and eukaryotic cell. You know, cell biology is a study of cells, their structure, function, as well as behavior. And all living things, they are made up of cells. They are having cells as a basic structural as well as functional unit, which are having extraordinary abilities to carry out all life activities at this level. And the simplest form of life, they consist of single cells known as unicellular organisms, while we know higher organisms, they are having billions as well as trillions of cells within a single body. And there exist some simple aggregations as well, which are known as colonial organisms. Here is some of the variety in shapes and sizes of the cells. Alpha diagram is आपके सामने नजर आ रही है, it's a single nerve cell from the mammalian brain and showing huge branching on all sides. Beta is the diagram of a picture of a paramecium, the protozoan, a single giant cell. It swims or carry out its movement with the help of cilia. Charlie is a chlamydomonas, which moves with the help of flagella, another significant form of algae showing locomotion as well. And diagram delta, it shows, D diagram, it shows yeast, Saccharomyces cerevisiae. This yeast cell used in baking bread reproduces itself by process called budding. And the last diagram is Heliobacter pylori. This bacterium is a causative agent of stomach ulcer. It's a picture showing different cells with a variety of shapes and locomotion sources. Cells, you know, they are the functional as well as structural unit of the life. They are the smallest independent units. And basically, cells are divided into two major types, prokaryotic and eukaryotic cells. Pro mean before and karyon mean nucleus. These are the cells which are most primitive, developed before the evolution of nucleus, and they are independent single-celled organisms such as bacteria and cyanobacteria without a definite nucleus. The other group, eukaryotes, they have a cell with membrane-bounded nucleus, और उनका जो nuclear material है DNA, उसको nuclear envelope cover कर रहा होता है, nuclear envelope surround कर रहा होता है. There are various other differences which will be discussed in the upcoming portions. But all cells are having basic three components: plasma membrane, the outermost boundary in animal cells, but it lies beneath the cell wall in case of fungi, bacteria, or plants. Cytoplasm, the area between cell membrane and the nucleus, it contains most of the organelles of the cell. Semi-fluid form of the cytoplasm is called cytosol, और उसमें जो suspended structures हैं वो क्या हैं? Organelles. The nucleus, usually known as control center, having genetic material in the form of chromosomes, is separated from the cytoplasm by its own nuclear envelope. And the nucleoplasm is the liquid portion, semi-fluid portion within the nucleus, just like cytosol in the cytoplasm. The next point is that why are cells usually mostly they are so small and can be seen with the help of microscopes. There are certain exceptions, जैसे हम जानते हैं eggs होते हैं cells के, fishes के, amphibian, reptiles, birds के, या कुछ longer nerve cells हैं, they can be viewed by the naked eye, but most of the cells, they are too small to be seen by the naked eye. तो उनको देखने के लिए हम usually microscope का सहारा ले रहे होते हैं, microscope use कर रहे होते हैं. One reason for the smallness of cells is that ratio of volume to the cell's nucleus, volume of the cell's nucleus to volume of its cytoplasm must not be so small that the nucleus, the cell's major control center, cannot control the cytoplasm. अगर सेल बहुत larger होंगे तो cytoplasm का एरिया इतना बड़ा हो सकता है that may not be controlled by the nucleus. That's why cells are so small. Another aspect is that limits the cell size is actually exchange of substances, exposed surface area. As the radius of the cell increases, cell volume increases more rapidly than the cell surface area. Because surface area तो वही होगा जो exposed area है, जबकि जो cell का volume है वो ज्यादा तेजी से increase कर रहा होगा. Just look at this diagram. Radius 1 centimeter है तो surface area है 12.5, volume 4.9. लेकिन जब रेडियस 2 सेंटीमीटर होता है तो वॉल्यूम 33 हो जाता है और सरफेस एरिया 50.26. ऑन द अदर हैंड अगर रेडियस 4 सेंटीमीटर है सरफेस एरिया चेक कीजिए 200 तक पहुंच गया जबकि वॉल्यूम 268. दैट मींस सरफेस एरिया इज नॉट रैपिडली इंक्रीजिंग एज द वॉल्यूम. दैट्स व्हाई सरफेस एरिया टू वॉल्यूम रेशियो उसको एक एक्सचेंजेबल लिमिट तक रखने के लिए कि सब्सटेंसेस को एक्सचेंज करना आसान हो. दैट्स व्हाई मोस्ट ऑफ द सेल्स दे आर स्मॉलर. ऐसा क्यों नहीं हो सकता कि एक सिंगल लार्ज सेल पे बॉडी कंसिस्ट करे रादर देन हैविंग सो मेनी स्मॉलर सेल्स? तो उसकी रीजन ये है कि स्मॉलर सेल्स इनका एक्सचेंजेबल सरफेस एरिया टू वॉल्यूम रेशियो या एक्सचेंजेबल एरिया इनका डिफरेंट पॉइंट्स पे सब्सटेंसेस एक्सचेंज हो सकेंगे जो कि इसको फिजिबल बनाएंगे, इजी बनाएंगे एक्सचेंज ऑफ सब्सटेंसेस को. There are some differences between typical prokaryotic and eukaryotic cell. Basic difference you know is the nucleus, which is absent in prokaryotic cell but present in eukaryotic cell. Then you can differentiate them on the basis of their other features such as membrane bounded organelles, which are not found, which are not present in case of eukaryotic cell, but they are, which are present in eukaryotic cells, but they are absent from prokaryotic. इसके अलावा इनका नंबर भी वेरी करता है. प्रोकैरियोटिक सेल्स दे आर हैविंग लेसर नंबर ऑफ ऑर्गेनेलिज एज कंपेयर्ड टू यूकैरियोट्स. यूकैरियोट के ऑर्गेनेलिज की टाइप्स, इनका नंबर, उसमें वैरायटी ज्यादा है. Furthermore, ribosomes they are smaller size 70s and attached as well as free in prokaryotes, but eukaryotes they may have 70s as well as 80s ribosomes which may be attached as well as free. प्रोकैरियोट के राइबोसोम स्मॉलर हैं जिनमें 70s का मतलब है कि लार्जर सब यूनिट 50s और स्मॉलर सब यूनिट 30 स्वेडबर्ग का होगा. जबकि यूकैरियोट के केस में लार्जर सब यूनिट 60s और स्मॉलर सब यूनिट 40s का होता है, जिसके अंदर फर्दर 5s RNA, 23s RNA in case of larger subunit of prokaryote and 16 Swedberg of ribosomal RNA in smaller subunit. In case of eukaryotic ribosomes, 5 Swedberg, 5.8 Swedberg and 20 Swedberg RNA is found in the larger subunit, while in case of smaller subunit, it is 18 Swedberg of the RNA. और इसमें आप लोग दूसरा जो पॉइंट डिस्कस कर रहे हैं दैट इज देयर अकरेंस आइदर देयर इन फ्री फॉर्म और अटैच्ड फॉर्म. एक्चुअली यूकैरियोट के अंदर तो आप लोग देख सकते हैं यूकैरियोटिक सेल में ये एंडोप्लाज्मिक रेटिकुलम के साथ भी अटैच्ड है और साइटोसोल में फ्रीली सस्पेंडेड राइबोसोम्स, फ्रीली डिसॉल्व्ड राइबोसोम्स भी नजर आ रहे होते हैं. पर इन केस ऑफ prokaryotic cells, they are found either dissolved form in the cytoplasm or they may be found attached with the plasma membrane. Sometimes they are found attached with the plasma membrane as well. That's why the next feature which we can discuss over here is presence of chromosomes as well as their structure. Chromosomes in case of prokaryotic cell, they are circular, single, as well as made up of DNA only. But as far as eukaryotic chromosomes are concerned, they are linear. There may be one or multiple chromosomes, but composition also varies. It is made up of DNA plus histone proteins. While in case of prokaryotic cell, the chromosomes which are circular, they are made up of DNA only. And you can see over here, histone proteins, they are found in case of eukaryotic cells only. The next feature which can be distinguished is the presence of plasmid. Plasmid, however, they can be found in both prokaryotic as well as eukaryotic cells, but prokaryotic cells, they are having it in cytoplasm, which are known as extra chromosomal DNA for having antibiotic resistance genes, and they are used as the vectors in biotechnology. In case of eukaryotic cells, mitochondria may have certain circular DNA as well as some extra chromosomal DNA as plasmid. And plants, they may have in chloroplast. Cell wall also varies if it is present in both on both sides. Eukaryotes, if they're having cell wall in case of plants, it is made up of cellulose, or fungal cell wall, aap log jante hain, it's made up of chitin. While prokaryotes, they're having a cell wall made up of peptidoglycan or murin, as you can see in this diagram. ये peptidoglycan या murin gram positive बैक्टीरिया में abundantly present होती है, जबकि gram negative में इसकी composition कम है. Cellulose or chitin. Chitin is found in as a major component in fungal cell wall, while in plants, the cell wall is having cellulose as its major component. Next difference can be discussed in their cell membrane. Cell membrane of prokaryotes, they lack cholesterol or sterols, and it is made up of phospholipid plus proteins only. While in case of eukaryotes, lipoprotein layer have additionally a component which is known as cholesterol, which is responsible for the stability as well as rigidity of eukaryotic cells. Prokaryotic cells, their cytoplasm shows gel-like consistency, while eukaryotes, they are having gel as well as sol-like portions. As far as cytoskeleton is concerned, prokaryotic cell is without microtubule or microtubule containing structures, while eukaryotes, they are having microtubules, intermediate filaments as well as microfilaments. While prokaryotes, they may have intermediate filaments or microfilaments only. But microtubules, they are usually missing in case of prokaryotic cells such as bacteria and eubacteria. As far as flagella are concerned, prokaryotic flagella, they are made up of flagellin protein, and these flagella, they run in rotatory movement. Prokaryotic flagella, they use a rotatory motor and proton driven pump which helps in the movement of flagella. While as far as eukaryotic flagella are concerned, they are made up of tubulin protein. As you can see, 9+2 arrangement of tubule is found, and these flagella, they show a bending movement rather than rotatory movement, and they are ATP driven rather than proton driven, and they show arrangement of microtubule in 9+2. By flagella of prokaryote, they do not have microtubules. They are made up of flagellin components or flagellin protein. The next difference can be considered in case of cilia, which are usually absent. They are absent from prokaryote, but usually present in eukaryotic cells, and they are having a similar arrangement as that of flagella, but they are smaller size and they are more numerous as compared to flagella if they are found as in case of eukaryotic cells. Pili, they are the features of prokaryotic cells only and used during conjugation or attachment, while eukaryotic cells, they are without pili, they are having other extensions. Size of prokaryotic cells is usually smaller, while eukaryotic cells as a whole, generally they are larger. As you can see in this diagram, mitochondria, which is an organelle of eukaryotic cell, is as the size of a bacterium, which is an independent cell. So eukaryotic cells usually larger होते हैं, जबकि prokaryotic cells, they are smaller. As far as their other features, functional features are concerned, prokaryote के केस में हम growth को define कर रहे होते हैं, increase in number of bacterial cells. As far as eukaryotic cells are concerned, it includes increase in number as well as increase in size of organism. Eukaryote में size और number of cells दोनों increase करेंगे, जबकि prokaryote में आप जो stages पढ़ते हैं bacterial growth curve की, इसमें सिर्फ number के increase करने को bacterial growth कहा जाता है. Cell division takes place with the help of binary fission in prokaryotes, while eukaryotes they carry mitosis and meiosis. And cellular respiration for the production of ATP in prokaryote is occurring within their cytoplasm. And their membrane is used as a source of chemiosmosis, site of chemiosmosis for ATP production. यूकैरियोट्स में प्रोसेस साइटोप्लाज्म के साथ साथ माइटोकॉन्ड्रिया में कंप्लीट हो रहा होता है. प्रोकैरियोट्स के अंदर जो फोटोसिंथेसिस का प्रोसेस है, इट विल टेक प्लेस इन सेल मेम्ब्रेन, साइटोप्लाज्म. इट मे बी सल्फर रिलीजिंग फोटोसिंथेसिस और ऑक्सीजन रिलीजिंग फोटोसिंथेसिस, और it may involve photosystem 2 only. While in case of eukaryotes, chloroplast, chloroplast is a site of photosynthesis. It's oxygen producing, and it may involve photosystem 1 as well as photosystem 2. So ये भी एक difference है. जबकि movement में flagella हो सकते हैं prokaryote में, non-motile, और eukaryote में variety of locomotion है with multiple organs for their movement as well as locomotion. डीएनए रेप्लिकेशन साइटोप्लाज्म में होगी प्रोकैरियोट्स के अंदर, यूकैरियोट्स के अंदर न्यूक्लियस में. ओकाज़की फ्रेगमेंट्स की लेंथ वेरी करेगी, और ओरिजिन साइट्स ये हम डिस्कस करेंगे इंशाल्लाह रेप्लिकेशन के टॉपिक में भी अलग से. ट्रांसक्रिप्शन में भी प्रोकैरियोट में सिंगल एंजाइम फेनोटाइप्स के आरएनए बना रहा होता है, यूकैरियोट्स में 3. फर्दर डिफरेंसेस विल बी डिस्कस्ड इन प्रोसेस टॉपिक ऑफ ट्रांसक्रिप्शन. साइट भी वेरी करेगी, यूकैरियोट में न्यूक्लियस है, प्रोकैरियोट में साइटोप्लाज्म. ट्रांसलेशन में साइटोप्लाज्म के अंदर प्रोकैरियोट में, यूकैरियोट में भी, बट द फर्स्ट अमीनो एसिड is modified in case of prokaryote for methionine, and eukaryotes they are having methionine. And it is a summary between differences between prokaryotic and eukaryotic cell. These are structural differences which we discussed one by one, and these are functional differences.